I'm Ruth Marshall. I'm a clinical associate professor at Adelaide Uni, running the rehab programs. And I'm delighted to be here today to introduce two other academics who I work with in rehabilitation. The first speaker this afternoon is Dr. Jillian Clark, Associate Professor Jillian Clark, who is going to speak to us about some of the research that we're undertaking here in Adelaide in spinal cord injury. There will be hopefully time for questions at the end, and I would be grateful if you put any questions into the Q&A area so that I can monitor them and then ask the questions on your behalf. After Jillian is finished, we will then hear from Associate Professor Anupam Gupta. Jillian started life as a physiotherapist, but hasn't done any physiotherapy for a very long time. I had the honour of being one of her supervisors for her PhD, and she's continued to go on with her interest in spinal cord injury research. So over to you, Jillian. Thank you.
Thank you, Ruth. Well, good afternoon, everybody. It's my pleasure to share with you today some snapshots from our spinal cord injury research program and to show some of the strategies that we have used to build research partnerships and capacity in this very specialised area and very difficult area in which to pursue as far as research is concerned. I'd like to start as well by acknowledging country and the original traditional owners of country on which we are meeting today and I'm talking. You can see that looking at the partnerships we formed, most of our partnerships arranged along the biomedical precinct of CALHN, SAMRI, U of A, with outreach and reach out, if you like, to Flinders Uni, as well as to the Eastern Seaboard, as well as international partnerships, mostly in the USA and Canada. When I refer to these, I refer to the topic of my conversation, which is bench-to-bedside translation. And I cannot possibly cover all of the work that we do right across the acute care, process of care to rehab in this talk. I'd just like to also acknowledge the leaders, the clinical leads, Prof. Brian Freeman, Mr. Wyatt Yao, Ruth Marshall, and some of the leads as well in the work that's allowed us to put together some stem cell biology, Stan Gronthos, along with smart biomaterials engineering, Jason Losik, as well as drug conjugates. Starting with spinal cord injury, there are approximately 400 new index cases in Australia each year. And of these traumatic cases, there are 1.26 per 100,000 population, as shown by Victorian work in 2019, and about 100,000 community dwelling Australians who are living with spinal cord injury. Ruth and I have done extensive work on lived experience and surveyed this in a 22-country partnership. And what we can tell you from that is that the lived experience does differ within and between individuals and within and between countries. But people with spinal cord injury describe major impacts on quality of life, health and well-being, social and financial equity. And this is not the topic today. I would like to focus on the urgent unmet need for viable therapeutics. And there's a number of drivers here. Some are compassionate, as well as the lifetime costs of care, and shown by Access Economics, quite old stats, 9.5 million for the lifetime of a tetraplegic person with tetraplegia, and 5 million for an individual with paraplegia. And for those who are not familiar with the topic, tetraplegia involves both the upper limbs, trunk, and the lower limbs, and therefore affects manual function, grasp, writing, personal care. Whereas paraplegia affects the lower limbs and torso, and in that case has lesser impact on mobility and personal care. Traumatic spinal cord injury presents as a medical emergency, and we can see here the derangement of the spinal column, the vertebra impinging on the spinal canal and compromising it. And we have, as well as that, spinal cord compression. And at the epicenter of this injury, we have a hemorrhage, and around the outside of the injury we'll have ischemic areas. And these are exacerbated by the very low blood pressure that we see in our patients at this time of the injury, very acutely and for the first week or so post-injury. There is a prescription around blood pressure to maintain perfusion of the cord and spare viable neurons at 85 to 95 millimeters of mercury. The data I'm showing here, it was taken from the triage data from the accident scene to the hospital. And here we can show very clearly that even a 10 millimeter increase in blood pressure in that period of time correlates very strongly with an increased likelihood of having some motor sparing at arrival. The things that we can't see here are the very severe cellular injury and the biochemical events. We know that we have disruption of axons from mechanical shear and disruption of vessels. But in addition to that, we also have cytotoxic injury. We have biochemical cascades which expand the injury beyond the epicenter. We have CSF flow obstruction above and below, so delivery of nutrients and substrate removal is also impaired. And all of these events culminate in cytotoxic or neurotoxic inflammation, which can be imaged using advanced imaging techniques. In relation to how we assemble a research program in spinal cord injury, at the very center of our hub is our clinical knowledge and expertise. And no translation from a bench to a bedside can occur without that clinical hub. We need knowledge of therapeutics. We've had experience in a number of therapeutics over the years, as well as regulatory controls and mechanisms. And we need access to diagnostics, and as well as that knowledge of delivery systems. And pulling all of those things together, we can begin to think about translational research. And the 3 research projects currently underway at the within CALHN, at the RA and across Hampstead Rehab Centre, tackle 3 of the major problems that we have in spinal cord injury, particularly in the acute phase: neuroinflammation, neuroprotection of semi-viable neurons, and neuroregeneration, the ability for to stimulate that limited capacity we have of the spinal cord to repair and regenerate. The first is looking at a radioligand. It is a translocator protein which has a very high affinity for receptor, which is expressed on the mitochondrial membrane. It's a marker of gliosis, or immune cell activation, the central immune cells in the cord. And this is imaged with PET-CT to give a readout of the inflammation and bring that back to diagnostics or therapeutics. In terms of neuroprotection, we're looking at riluzole. This is a drug which has shown efficacy in motor neuron disease to increase ventilator-free days. And we're now laterally translating that to spinal cord injury to see its effect on motor recovery. Unfortunately, this trial was ceased during the COVID outbreak, and there are now some uncertainties about its sponsorship. And this is by no means an isolated case. It's a universal thing, both in our centre and in other centres overseas. In terms of regeneration, we look at a monoclonal antibody. As mentioned earlier, the spinal cord has very limited capacity for repair and to regenerate. And this is targeted to regeneration. It's blocking a molecule which blocks the spontaneous capacity repair and does so to stop aberrant connections. Coming back to the bench, the change of pace, I'd like to introduce you to our work on innate host defense. This is the very first cellular event to injury, the way the body begins to protect itself, and it can be both beneficial and harmful. We know that cells from the peripheral blood are recruited to the spinal cord, and intriguingly, they are able to cross the leptomeninges and are attracted by the remote brain choreplexus and travel down with the CSF to the injury site. And they can be either neurotoxic or promote regeneration. And clearly, it's important to phenotype these cells so that we can better utilize them for therapeutics and diagnostics. Returning to the slide earlier on the clinical trial, this is a preclinical data from that same from that work from Ryan Doig's lab in SAMRI. And you can see here the area that is injured in the rat model of spinal cord injury at the 10th thoracic vertebra. We have here normal healthy control animals. We have laminectomy control animals, or sham as I'll call them. We also have laminectomy plus spinal cord injury. Looking at the 24 to 48 hour readout, we see very clearly we are getting a readout from our radioligand to this receptor in our animal model. And when that's plotted, it's very clear there's a significant increase in uptake in the spinal cord injured animals in relation to the sham and the controls. And this is, it does persist, but does actually decline over time. This is equivalent to gliosis, it's a marker of gliosis, and has been also studied in other conditions. And thinking about inflammation, this has a role not just in the injury of the cord, but also in the pathology of neuropathic pain, which is a common complication of spinal cord injury. And in this respect, we were able to obtain Department of Defence funding to examine the pain profiles of our patients, and we found that 75% of males reported they had chronic persistent debilitating pain after their injury, and this only 35% of females reported chronic persistent pain, and the reason for this is unclear. We went on to look at opioid exposure. This is a drug which has been shown to activate inflammatory pathways by Mark Hutchinson's group at the U of A in paradigm shifting work about chronic pain and a range of chronic pain conditions. And when we looked at opioid exposure at the time of injury, we saw that the patients who had pain had had higher doses of opioids, and this was significant at the 0.02 level. And it did appear that the opioids were antagonizing the propensity for pain in this group. This wasn't found for other opioids, so not for fentanyl or for codeine. We found that amitriptyline had a countering effect and was beneficial in this regard. I went on to genotype the patients with Janet Collar and looking at the prototypical polymorphisms associated with the inflammatory pathway, starting with toll-like receptors, looking at mu opioid receptors, and looking at the inflammatory readout of interleukin-1 beta. And we did find a very significant genetic signature which correlated with propensity for pain together with exposure to opioids in that study. Shifting back to the bench again and thinking about the peripheral blood cells and which are being recruited through the coronal plexus and infiltrating the injury site, we took peripheral blood samples from our preclinical model in the rats at SAMRI, and this was work from Sandra Jenkins' PhD. Sandra was able to identify a very novel subset of monocytes in the peripheral blood. And this was a transitional population with very low monocyte maturity. And this population was depleted in our spinal cord injury model. She went on to take a look at how peripheral blood monocytes, when co-cultured with dental pulp stem cells, might modulate those cells. And she did that under conditions that were cytotoxic. And looking at the spinal cord injury data here in blue, Sandra was able to show that these cells can modulate the stem cells to anti-inflammatory phenotype, suggesting that this is indeed potentially helpful and maybe a potential therapeutic. We collaborated with colleagues at Flinders, James Patterson and Jim Mitchell, to further characterise this immune signature. We looked at viral load initially, and this is our first readout, and we did not get a strong hit on that. The reason for looking at virus is that microbiota from the gut carry a number of viral pathogens. They are colonised, and with spinal cord injury, there's a theory that the release of microbiota to the peripheral blood may carry with it risk of viral load. What we did find, and I am not at liberty to show you, is a very unique signature, which we are now running against purified exosomes for a readout.
Jillian, you've got about 30 seconds left.
Right. Coming back to the bedside, we now have identified very interesting and new tools to look at our finding that peripheral blood immune cells are able to predict the likelihood of recovery in a patient. We've gone on to partner some of our dental pop stem cell work with smart biomaterials, showing that we can actually improve the biodegradability of smart biomaterials with composite materials, and we can culture at high densities stem cells on these scaffolds. And finally, I'd just like to mention some work in drug conjugates carried by a gold nanoparticle, showing retrograde transport through the nerves to the brain and the cord. Now, these gold nanoparticles are cytotoxic, and after a conversation with Guangzhou Mao, we've now collaborated with Guangzhou to bring our clinical knowledge to this work with new drug conjugates. I hope that I've been able to give you some snapshot of our research capability and how we've pulled together a bench-to-bedside program in a very, very short period of time, just a few years. I'd like to acknowledge in particular Stan Gronthos and Ryan Doig, our clinical leads, who I acknowledged earlier, Mark Hutchison and Janet Collar, Guangzhou Mao for the drug conjugates, and Dusan Lusik for biomaterial work. And I'd like to also thank our sponsors. Thank you for your attention.
Yeah, thank you, Jillian. There aren't any questions, and we'll move straight on to Dr. Anupam Gupta. Anupam is a clinical associate professor, again at Adelaide University, and has an interest again in a wide range of rehabilitation associated research, including picking up errors and also spasticity. So I'm really pleased to hand over now to Anupam. Thanks, Anupam.
All right, so I'll start. I'm Dr. Anupam Gupta, so I'll present this on a very difficult problem I encountered in a group of patients who had developed a kind of hand ulceration caused by focal spasticity. And human hand is has a very complex anatomy with a lot of bones and muscles, and we have multiple grips and postures from a mobile thumb. And we all know the hand is a multi-purpose organ for reaching, grasping, manipulation, and feeling. And thumb has great mobility, and a lot of our superiority in the animal kingdom can be ascribed to our mobile thumb. And hand is an important conduit to our reach, if not infinite, tactile, social, and communicating world. The evolution of the hand became possible to a large extent by the highly mobile thumb. Hands makes us productive and human. So if you look at this slide, this is a Nature paper. This paper compares humans with chimpanzees, and we have a bigger thumb compared to our hand. And if we compare our hands with the hands of a chimpanzee, and hands have attracted human imaginations, and this is how the human hand has evolved: shorter thumb, longer thumb, and we can oppose which the animals cannot. And then, it is, quite known that prehistoric humans painted hands. The left hand is a painting from Chauvet Cave, and right one is the Aboriginal Australian painting about 60,000 years old. So a lot of neurological condition gives rise to spastic hand, where the thumb loses its mobility and gets stuck to the palm, which is known as thumb in hand deformity, and fingers flex unbidden due to hyperexcitability of the stretch reflex, and hand can no longer perform its function. And then these are the conditions like stroke, cerebral palsy, multiple sclerosis, brain injury, and surprisingly 73% of the nursing home residents has central nervous system disorders. So in the spastic hand, these spastic fingers dig deep into the palm and give rise to palmar ulceration, which is very difficult to treat, and it is a terrible condition in terms of human suffering and in terms of carer burden. And in this slide, I have shown the areas where they can develop the ulceration, the areas marked red, and I'll show you some of the hands. This lady was in a nursing home with spastic hand, and if you open the hand, besides this grotesque deformities, there was ulceration of the hand. So the hand looks like that. And then when they develop spasticity, the nails dig into the palm, and that's how they develop the ulcerations. This is another post-stroke hand. So the consequences are terrible because the hand smells offensive. The carers cannot clean the hand, and they develop atypical pressure ulcerations. They're pressure ulcerations, they're called atypical because they're in not the typical areas of pressure ulceration. So from a highly evolved organ, the hand becomes a source of pain and suffering. So these are some of the hands. So we took up these patients. They came to our clinics, and we took up these patients and we tried to treat them because the spasticity was the main cause of these deformities. So we thought we should treat the spasticity first. So they had treatment-resistant, long-standing hand ulcerations. The caregivers were struggling with it. So we took up their case, and we first treated them with toxin, and we followed them with some selective splinting and therapy. And to our surprises, all of them responded very well, and we produced 2 papers out of it. And you can see the hands, how they look like. They sweat and they develop debris, and they smell terrible, causing intense suffering. And some of the patients even didn't allow the caregivers to clean their hands. So maintenance of hygiene was difficult. That's another hand. This is the interphalangeal joint clefts, and the metatarsal joint, phalangeal joint areas were involved with ulcers. Normally, all of them have, this is the hand area, if you look at their brain imaging. So we took up these patients for treatment. They can have ulcerations around their elbow and in their axilla as well. So they are commonly confused with another condition called Dupuytren's condition, which does not have any upper motor cause. So they are a different group of patients altogether. So we first decided to tone down the spasticity with toxin, and then we splinted them and we maintained the hygiene. We identified the involved muscles. There is you can identify very precisely even a lumbrical, as I am showing from this short video. And then we treated them with sheepskin splints, and then we used these bread flex tubes to stretch their fingers, followed by thermoplastic splints. So most of the patients got healed. We came out with some ideas of how we measure the outcomes pre and post. So we measured the fingertip to palm distance when they came, and at the end of the treatment, as you can see, because that gives us an objective outcome assessment, and followed by different kinds of splinting. So we measured their muscle tone. On examination, one has to look for the muscle tone, power, reflexes, temperature, hyperhidrosis, and bromhidrosis or offensive smell. So these are the data sheet, how we kept our records, the muscles we injected, names of the patients, and these are the different parameters. So these were extremely painful. So we recorded their visual analog scale. We graded their pressure ulcers from 0 to 4. We measured the fingertip to palm distance. We measured the carer burden scales and gas goals. So they are showing pre and post measurements, and by all parameters, there was significant improvement. As you can see, p values were less than 0.002. So, and some of the pre-post pictures I'll show you. You can see the picture on the left side that proximal palmar crease ulceration has completely healed. The distal ulceration hasn't healed yet. He came for a second injection, but then after that, it was completely healed. This is again another pre-post photographs. And look at this one with this hand after injection and splinting became the hand on the right-hand side. This hand also had remarkable results, and we were surprised. Some of the patients regained some of their hand function as well. So then we looked at the different mechanisms. There are different mechanisms by which we got this results. So there were, first thing is toxin release reduces the muscle contraction and spasticity, but it causes smooth muscle arterial vasodilatation, and then there were effects of toxin in pain. It is quite interesting that now toxin is being tried in conditions like as Raynaud's phenomenon or vasospastic disorders, and in some neuropathic pain. I'll come to that. So I have used these graphics to give you an idea. So toxin now people think that a lot of research is coming out that it could be helpful for pain because it reduces peripheral sensitization by depleting the mediators of inflammation and mediators of pain, and there is retrograde transport of toxin, so it reduces peripheral and central sensitization, and that's how it can cause relief of pain. Very interesting effects. We have done a systematic review we will publish soon on this topic. And then excessive sweating. The toxin actually inhibits sympathetic fiber signal to the eccrine glands and thereby reduces sweating, because when there is excessive sweating and debris built up, the ulcers takes long time to heal. And then effects of vasodilatation. So toxin actually impedes secretions of acetylcholine, and it sends signal to acetylcholine mediated arterial muscles, and it causes paralysis of the acetylcholine mediated arterial vasoconstriction. It causes vasodilatation, and that possibly improves ulceration when there is greater supply of blood. Naturally the ulcers heal. So we published this work. One came out in the Medical Journal of Australia as a lesson from practice, and then we published the series in International Wound Journal. And in that journal, we argued that a randomized controlled trial is unethical because the results are so remarkable. So if it is giving such good results, why should we randomize people to saline injections, so on and so forth? I was surprised that the editor kept that line and published the article in their journal. So this is our clinic, members. I would like to thank them for their help, and thank you all. And I'm happy to take any questions.
Thank you, Anupam. Is there a need for funding for this sort of research that you see? Because obviously at the moment, the issue is that they're saying, well, it doesn't need to be a controlled trial because the efficacy is so clear. So what are your thoughts about that? Can we extend this into the research area?
Okay, so thanks for this very good question. So also now we have got the PBS approval, so this is part of our service. But as I have indicated that the toxin may be useful in neuropathic pain. There are some RCTs coming out, and there is a condition we deal with post-stroke pain. The results are remarkable. So if we want to establish toxin as an effective treatment for neuropathic pain in certain conditions, definitely we would need the funding. So we have just completed a systematic review and meta-analysis. It is under press. So once that is published, we then apply, because we cannot do that study without funding, Ruth.
Okay, thank you.
Anupam, can I ask a question, Ruth? Still in.
Yes.
Anupam, I was interested in, it was a comment really. We've used many years ago in our rat models, TET-tox as a retrograde, to map retrograde transport in sensory and motor neurons and through the spinal cord into the brain. And it seems to me that you are describing a very similar mechanism and a way to access the central nervous system that avoids the blood-brain barrier, which is also the focus of the last slide that I showed for the nano-drug conjugates, which are similarly transported.
Yeah.
I think the idea of it being an access route is very interesting, and you probably need to think about, if our patients are receiving it, and particularly our cord patients are receiving it, any potential off-target effects for treatment of spasticity with botulinum toxin in that group.
Yes, so there are some very good studies, preclinical studies, animal studies, where they have established that the toxin transport in a retrograde way, because I have seen, because remember, when I treated these hands, I didn't inject their palm. I injected the specific muscles which are in the forearm. But yes, it is established that the toxin spreads through the, through the axon, and that's how they think that it relieves the pain. Because if in order to get a relief of pain, that's how you can explain. But I think with different methods, they have now shown that they retrogradely, they get transported to the spinal cord, but there were no indications that they had any adverse effects from them, because they are very tiny amounts,
Yes.
Indeed.
I'd like you all to thank our two speakers who are showing you some of the other type of research that is going on throughout CALHN, both at the clinical and at the translational level. So thank you, Jillian, and thank you, Anupam, for presenting today. Thank you all.
Thanks.
My pleasure. Thank you, Ruth.